Hello friends, welcome to Veterinarian. So today's topic is oxidative phosphorylation and uncouplers of oxidative phosphorylation. First about the oxidative phosphorylation. Let's talk about its definition. What is its definition? It is a process of coupling of oxidation of the NADH or FADH2 in the electron transport chain and phosphorylation of ADP to form the ATP in the electron transport chain. So why it is called oxidative phosphorylation? Because there is two processes. First one is the oxidation and the second is the phosphorylation. So when the electrons flow from the NADH or FADH through the electron transport chain, they release free energy. The part of this energy is used to generate ATP and the other part is used to generate the heat. So ATP is generated, which is known as the phosphorylation. Other is used to generate the heat, which maintains the body temperature. Okay, now let's look on to the mechanism of oxidative phosphorylation. So there is flow of electrons from the NADH or FADH2 in electron transport chain is not directly linked with the ATP synthesis. Why? Because it is a complex process and several hypotheses have been put forward to explain this process. Among them, the chemiosmotic hypothesis, which is explained by Michel, is the most accepted one. So the hypothesis which explained by Michel is the most accepted one. So we can look on to its hypothesis. So next topic is chemiosmotic hypothesis or Michel's hypothesis. Why it is called Michel's hypothesis? Because it is coined by Michel or it is explained by Michel. So first point is, when electrons flows through this electron transport chain, free energy is released. We can look onto this diagram. So this is the mitochondria. This is the matrix. This one is the inner mitochondrial membrane, and this part is the intermembrane space. So matrix, inner mitochondrial membrane and the intermembrane space. So the complexes 1, 3, 4 as well as 5, these are present on the inner mitochondrial membrane. So we can look on to this hypothesis. So when electrons flows through this electron transport chain, so when the electrons, there is electron which flows through these channels, free energy is released. With this free energy, protons which are present on the matrix traverses through the which part? Through the complexes and reaches the intermembrane space. So it reaches the intermembrane space. So it's the pumping of protons from the mitochondrial matrix to the intermembrane space. So which act as a proton pumps? Complex 1, complex 3, complex 4 of the electron transport chain. These protons which are transported from the matrix to the inner mitochondrial membrane cannot diffuse back. It cannot diffuse back into the matrix. Why? Because the inner mitochondrial membrane is impermeable to protons. So the inner mitochondrial membrane is impermeable to protons. So what will happen? There will be an accumulation of proton in the intermembrane space. So there will be more protons in the intermembrane space as compared to that of the matrix. So what happens? There will be a generation of a proton gradient because there is difference in the concentration of the protons. So it generates a proton motive force. So a proton motive force is generated. A proton motive force is generated as a result of the proton gradient. So the protons that accumulated in the intermembrane space, that is these protons, can flow back to the mitochondrial matrix only through the complex 5. So this complex is called as the F0 F1 complex or the ATP synthase complex or the complex 5. So the proton motive force is generated. So the proton motive force which is generated as a result of the proton gradient. That is the proton gradient results in the synthesis of ATP. ATP synthesis from ADP and inorganic phosphate by the enzyme ATP synthetase. So let's now look on to the core topic, that is the ATP synthase complex. This is the ATP synthase complex or the complex 5. It is present on the inner mitochondrial membrane. here. It is present on the inner mitochondrial membrane. It consists of two subunits, F0 and F1. It consists of two subunits, F0 and F1. This is the F1, and this is the F0, and this is the F1. Here 0, here 1. So F0, it spans the membrane and forms a proton channel because it helps in the entry of H plus from the intermembrane space to the matrix. So it is why it forms the proton channel. And the F1, it helps in the phosphorylation and it projects into the matrix from the membrane, inner mitochondrial membrane. It projects into the matrix. So F0 spans the membrane and forms the proton channel. F1 projects into the matrix and the phosphorylation mechanism. So there will be a flow of protons through the F0, which causes a rotation of F0. There will be change in the F0, which leads to the formation of ATP in the F1 subunit. So when H plus flows through it, there will be a change in the F0. There will be rotation. There will be a rotation of F0, which results in the conversion of ADP into ATP. Okay. So from where the energy is derived? The energy is derived from the proton motive force. This proton motive force generates energy, and the proton motive force is due to the proton gradient. Now let's talk about the inhibitors of oxidative phosphorylation. We know that the complex phi consists of two parts, that is the F0 part and the F1 part. F0 complex or the F1 complex. So there is F0 inhibitors and F1 inhibitors. F0 inhibitors, that is the ATP-ADP transporter inhibitor. Example is atractyloside. And about the F1, that is the ATP synthase inhibitor, is the oligomycin, which is an antibiotic. So here there is O and here there is O. So opposite O. If it is F1, then it is oligomycin, that is an antibiotic. If it is F0, it is not O. It is not O, that is atractyloside. Now we can look on to the next topic, that is the uncouplers of oxidative phosphorylation. Why the term uncouplers? Because it doesn't couple, which means it separates. Uncoupling means separates. Uncoupling means separates. So it separates the two processes, that is the oxidation process in the ETC and the phosphorylation process. So the uncouplers act as a proton channel in the inner mitochondrial membrane. So here is the inner mitochondrial membrane. So the uncouplers act as a proton channels, which helps in the it helps in the transport of H plus from the inner mitochondrial membrane into the matrix. So what happens? So it causes the leakage of proton back to the mitochondrial matrix. So the proton gradient is not present. So the proton gradient is not present. So the proton motive force is not generated. Since the proton gradient is not present, so the proton motive force is also not generated. So the ATP synthesis is inhibited. So the uncouplers, which allows the oxidation to proceed but not the phosphorylation. The uncouplers allows the oxidation to proceed but not the phosphorylation. The energy produced by the electron transport in ETC, because due to oxidation, energy will be released. Part of this energy is used for phosphorylation. And part is used to generate heat. This heat maintains the body temperature. So since here the energy which is required from the oxidation is only going to generate the heat, that is the body temperature. So the body temperature will be increased. In this case, body temperature will get increased because majority of the heat is used to generate the body heat, because the phosphorylation process is not taking place. Now let's talk about the ionophores. What is meant by ionophores? These are lipophilic substances. That promote the transport of ions across the biological membranes, which means it is uncouplers. So all the uncouplers are ionophores because these are lipophilic substances and it promotes the transport of ions across the biological membrane, that is from the intermembrane space back to the mitochondrial matrix, thereby preventing the proton gradient. Now we can look on to the examples of ionophores or examples of uncouplers. First one is the 2,4-dinitrophenol, 2,4-dinitrocresol, dicumarol. This allows the passage of this cation, that is H plus, through the mitochondrial membrane to the mitochondrial membrane, from the intermembrane space to the mitochondrial matrix. So it dissipates the proton gradient. So proton gradient is not a generator. Next example is thermogenin. It is a protein which is present in the mitochondria of brown adipose tissues, that is in newborns as well as in hibernating animals. So next example is thyroxine, which is a physiological uncoupler. Then some antibiotics. Some antibiotics also act as uncouplers. Example, gramicidin A, valinomycin and nigercin. So examples are 2,4-dinitrophenol, 2,4-dinitrocresol, dicumarol, thermogenin, thyroxine, as well as antibiotics. Antibiotics like gramicidin A, valinomycin, nigercin. Thank you.